Hello, and thank you for joining us. You are about to watch an impactful webinar on the relationship between genetics and cardiomyopathy. Presenting his experience will be the distinguished Professor Hendrik Milting, Director of the Erich and Hannah Klessmann Institute for Cardiovascular Research and Development at the University Clinic of the Ruhr-University Bochum in Germany. We will begin with a short background of cardiogenetics and spend the rest of our time describing some fascinating case studies from the Institute. Now, without further ado, I'll turn it over to Professor Milting.
Thank you, Sean, for the introduction, and hello, everybody. So I will present some data on the impact of next-generation sequencing in terminal heart failure, which we did here in Bad Oeynhausen, which is a big transplantation unit, as you will see in the next slides. So these are my disclaimers and disclosures. And this is the overview on the webinar. So I give you a short introduction on the background of our activities. I will come to some aspects of terminal heart failure and genetics, and I will show you some things of the practical in practical terms, how we do the diagnosis and the algorithms which we use. And I will also go in detail on the advantages and limitations of NGS in cardiogenetics, and then I will present what will be quite interesting to you, the case reports, which might be some surprising cases which you might also be confronted with in the clinics. And we'll end up with the conclusions. Okay, this is the background where I'm sitting currently is in Bad Oeynhausen. This is in Westphalia in Germany. And as you can see here on the left side, this is the registry for heart transplantations in Germany. And as you can see at the top of this, this is Bad Oeynhausen, which is really a huge transplantation unit and the #1 in Germany for decades in the meantime. Well, on the right side, you see a typical, I must say, a typical pedigree of a patient which we transplanted, which is highlighted by red color. So we transplanted this patient because he had a terminal heart failure and right ventricular cardiomyopathy. And what you can see also is that the blue ones, so his brothers and his nephews, were also affected. So we didn't see the family, we have seen the index patient, but obviously there is a family problem, and this might be caused, or this is of course highly probable that there is a genetics behind this kind of cardiomyopathy. Well, coming to transplant, back to transplant, as you can see here, this is taken from the International Society for Heart and Lung Transplantation. And what you can see here for two different periods, the amount or the proportion of patients with different indications for heart transplantation. So the yellow ones are the myopathies. In other words, they are the majority of cases who had to be transplanted, whereas the coronary artery disease is a incomparably smaller group of patients with need for heart transplantation. Well, so in other words, there's a huge amount of cases with terminal heart failure among the transplant candidates, and this is a motivation to go into more detail to find out what is the reason for their heart transplantation. The other reason to do genetics with these kind of patients is shown here on this slide. You see an article from the Yellow Press on the right side. I'm a woman without her heart. This was a spectacular case here in our hospital. And as you can see on the left side, this patient was transplanted aged 15 years old. And she rejected her donor heart around about 6 years later, the day before Christmas, under special conditions, I must say. And she was rescued by the implantation of a total artificial heart. So the donor heart was removed, and she received a donor heart. At that time, this was a ventricular assist device not designed to be a total artificial heart, but the surgeons here in our hospital managed to construct a total artificial heart from these devices. You can see the theaters here from the operating room. Well, What you also can see from the pedigree is she's not the only one in this family, and obviously, finally she died 1 year later due to complications under very bad conditions. And what you can see here is the whole family received a kind of post-traumatic stress syndrome. So in other words, there's a need for clearing up, for finding out what is the reason for this disastrous situation. And the question is, what about the sister of the index patient? Is she in danger to receive the same disease? What will happen to them? So there's a motivation that that patients are asking for that, asking for genetics for the reasons, and they especially the terminal heart failure patient in our experience are highly interested to find out what is the reason for their situation. Well, coming back to genetics, you see here on this slide different forms of cardiomyopathies: dilated cardiomyopathy, hypertrophic cardiomyopathy, HCM, and ARVC or arrhythmogenic cardiomyopathy at the bottom. And all these different phenotypes have a different estimated amount of genetics behind that. And especially when coming to dilated cardiomyopathy, which is the main indication for heart transplantation, it's estimated in the literature between 35 to 50% of the cases might have a genetics, but exactly checked was this in almost not in the transplantation units here, especially in Germany, which I know. Okay, how can you identify that a patient is affected by a genetic disease? And again, the pedigree I showed you before, you will obviously see that this might be a familial disease, but look at this pedigree. Would you expect that this family has a genetic problem? Well, just to sum up that at this position, at this point, at this time, of course, this pedigree below is also a genetic disease, and I will show you this example and why we are quite sure that this is a familial or a genetic disease without a familial phenotype. Well, most of these genes which are affected by mutations are listed here. Just to give you a brief overview, you will find this in the literature, of course, in several papers, reviews, etc. And there was a recent issue from circulation research with excellent overview of different cardiomyopathies. Basically, genes are affected which are responsible for the coding of proteins from the sarcomere. From the surface, plasma membrane proteins, but also from the nuclear lamina. There are also some striking examples of nuclear lamina proteins which are affected by mutations. So this is what we are doing. We have our clinical activities in the center, of course, and what we are doing in addition is, since more than 18 years, we are collecting explanted hearts in a biobank. So we have both. We have the blood, we have the patient, we have the blood from the patient, and we have also the myocardium. And we are doing molecular research on the mechanisms of the disease. And this is part of the genetic counseling activities we have. And of course, because we have this experience, this is part of the diagnostics we're using in the meantime for treating or for diagnosing our patients. Well, some technical issues. We started, of course, as others did as well, using Sanger sequencing. This is a striking example where we found some remarkable mistakes Sanger sequencing can cause. What you see here is data from a cell culture, which might be not impressing because it's not directly tissue from the patient, but it's remarkable what we found for proving the genotype of iPS-derived cardiomyocytes. What you see is at the top an indication by the arrow that there might be a homozygous mutation in the gene TMEM43, which is a very deleterious mutation, and which is the last example of my presentation in this webinar. At the bottom you see what is the real phenotype, or the real genotype, sorry. You see that this is a heterozygous mutation, not a homozygous mutation. So obviously by repeating this, we found that this was just a Sanger artifact, which cannot be explained by contamination with wild-type DNA, because this mutation is really, really rare. So in other words, this is cannot be explained by contamination of the Sanger sequencing. So Sanger sequencing in our hands made several mistakes which we never found up to now with NGS sequencing. Well, you see here a limitation of NGS on the other side. This is a good example for detecting a heterozygous deletion in the gene MORC. You can see that the coverage of the sequencing and the BAM file was dropping significantly down, and this indicates that there is obviously a kind of deletion present in that gene. But unfortunately, this cannot be guaranteed for every sequencing reaction which is realized by panel sequencing. So you should be aware that NGS does not really cover large deletions, chromosomal deletions. This should be checked by other methods in addition. But anyway, we're using this for sequencing in our transplant patients, and this is the workflow. We are sequencing these data by panel sequencing. We are using the cardiovascular genes, the panel from Illumina currently, routinely. And of course, this is first checked against controlled genomes, preferably of the exact server, of course, looking for the prevalence versus allele frequency. Then we are checking all the other sources. You will probably also use databases, Medline. We're looking for in vitro evidence, and I will show you some examples from in vitro evidence. So for functional data, and we are looking also for computer algorithms and silico evidence, etc. Then we are going back to the family, looking for co-segregation and the inheritance. We are looking for pathological evidence by the biopsy. We have always a pathological examination for myocarditis in addition. And finally, we end up with a classification which we are using is taken from the ACMG standards and guidelines, resulting in a classification starting from 1 to 5, and 5 and 4 is used in our hospital, especially to recommend, for instance, also predictive testing if this is asked by the family. So, but we should consider, for those of you who are not familiar with human genetics, a mutation might be pathogenic, ACMG5, but might have an incomplete penetrance. So in other words, you might have also older people, older carriers of that mutation who did not develop the phenotype. A pathogenic mutation might have heterogeneous expressivity, and I will show you a remarkable example for that. So you might be confronted with different cardiomyopathies caused probably by the identical mutation. And finally, a genetic disease might be caused by more than one mutation, and pathogenetic evidence versus clinical evidence is also, of course, sometimes an issue which has to be considered when you are doing genetic counseling in the clinical setting. Well, we start with the first example, and you might recall that this, I showed you this pedigree before. This is a case of a 6-month-old boy receiving a biventricular assist device, a Berlin heart, as you can see here on the right side at the bottom. And this young boy had a restrictive cardiomyopathy, which is a rare event for heart transplantation candidates and rare indication for ventricular assist device cases, but not so rare in our hands in the pediatric hospital we have here as well. Well, what you can see is the family has no other cases of known cardiomyopathies or myopathy, as we found out by interview of the parents. And in addition, a recessive pedigree was also not very highly probable because these parents came from completely different areas in Europe, so there must be not a genetic connection between these parents. So in other words, we were confronted with a very severe case of cardiomyopathy, but obviously not a familiar case. Of course, this might be a myocarditis, but is that really true? So we started panel sequencing with this kit, and we found a mutation in the troponin I gene, TNNI3, and the exchange was aspartate 127 against tyrosine. And as you can see here from the mammals, this is as the rest of the molecule is a highly conserved residue of TNI. Well, just to recall that the troponin complex is very important to move the tropomyosin from the thin filament, and this allows the thick filaments, the myosin heads, to interact with the binding site on the actin filament, and this starts the contraction. And just to remind you on that issue, the troponin I is responsible to move that, and the mutation was found in a heterozygous fashion in that index patient. In other words, about 50% of the troponin complexes here distributed over the thin filament carry the mutation, whereas the others are the wild types. So in the same filament you might be confronted, or on the molecular level you are confronted with a situation where this troponin I does not work in a proper manner, whereas the other one acts like the wild type does. So just to recall that, troponin of course is also used, as you can see here in the plasma, as an infarction marker, biomarker, but of course this is not relevant for what we are doing here with the genetics. So this is the data taken from Cornelia Jacquet. Cornelia is a colleague of mine from the same faculty who is interested in troponin, and she introduced this mutation. What she found was that this troponin I has a severely impaired activity concerning the binding to immobilized tropomyosin. And what you also can see here is that the binding activities to actin are also significantly changed. So in other words, we had in vitro data which indicate that indeed this mutation is harmful for the patient and might be the leading indication for this disease. Well, coming back to the pedigree, and when it came to genetic counseling, we were confronted with a special situation because the mother was pregnant for a second boy, and you can see here all the genotyping we did. So today we know that indeed this appeared to be a spontaneous, a de novo mutation in the affected kid, who unfortunately died by a device failure of the ventricular assist device. So the patient died, and the mother came to genetic counseling, was pregnant again, and she was highly interested to find out is the second child also affected by that mutation. And it is not, and as you can see also, the parents are not carrier of that mutation, but there was a small risk for being a genetic mosaic in one of the parents, so the parents decided to make a prenatal genetic testing of that kid, of that second child, and the second child didn't carry the mutation. And this is 4 or 5 years before our time. So in other words, this kid is healthy, is doing fine, etc. So why was this child not born with a cardiomyopathy? Well, there might be also an explanation for that, because the troponin I, this isoform, the cardiac isoform, is switched on at birth. So in other words, during pregnancy, this child has a skeletal muscle version of the troponin, which is not causing this kind of molecular damage in the sarcomeres. Well, we had a second child actually with a troponin C mutation, And this was born in the same situation, a de novo mutation in the parents, and this patient was directly transplanted, but this kid was born with a cardiomyopathy, and the explanation from the molecular point of view is troponin C is also present in the myocardium before birth of the child. And this might explain the different cause of and the different onset of these two different troponin mutations. Well, coming to the second set of example, or to other examples here, this is a typical example of a right ventricular cardiomyopathy. As you can see here on the right side, you see only some islets of myocardium in the right ventricle. You can see here the forceps and my fingers here. And you see, this is taken from the left chart, the papillary muscles of the left ventricle and the mitral valve here. So you see that fibrosis is also present already here in the left ventricle, and you see also some scarring in the septum. The right ventricle is almost completely white from fibrotic tissue. So the background for this mutation is a PKP2 mutation, a typical mutation, plakophilin 2 mutation, a nonsense mutation, which is typical for these kind of genetic disease. So this is a histological feature of that heart. You see a huge amount of fatty tissue, which is also a leading indication for the pathologist when he or she becomes a biopsy. Well, what is known about ARVC, especially for the classical forms, is that the intercalated discs, which are separating the cardiomyocytes, are ruptured because when there is in the connecting desmosomes, when there are some mutations in the affected proteins. This is the desmosome again, and what you see here is that these desmosomes are connected mainly by the catechoreins, desmoglein 2 and desmocholine 2. But on the other side, on the intracellular side, there is desmoplakin, plakoglobin, plakophilin, and intermediate filaments as well, which might carry mutations for this kind of phenotype, for this kind of cardiomyopathy. And all of these are classical genes which should be checked when you are confronted with a ARVC, the classical ones. But I come back to this later with another example where you find some heterogeneous expressivity with a certain mutation, which might also lead to an ARVC phenotype. So this is what is taken from the clinicians, from my clinical colleagues. They are using this for diagnosing the clinical phenotype. The task force criteria are used, and you see that you have some special things to go through to this table just to find out what is really how valid is this diagnosis. This is another example, again a pedigree, and as you can easily appreciate, there's no other case in that family. Some guesses that there might be a disease, but this is definitely not the case. So this patient was again checked by sequencing, and we found out that this patient is a carrier of a mutation in desmin. And this desmin mutation is unusual because the patient was a ARVC patient, not a patient not a patient presenting with a dilated cardiomyopathy or with a skeletal muscle myopathy, as you can see here from other known mutations in that gene. So this was a surprise for us because the N116S mutation was a novel mutation for us causing ARVC. So N116S is a mutation which is affecting a very strictly conserved motif in the intermediate filaments desmin is belonging to. You see this LNDR motif is also present other family members from intermediate filaments. So vimentin, these are different keratins, lamines, urofilament, and the asparagine residue here is present here. In all these different family members. And this is also highly conserved in a phylogenetic sense in Hydra vulgaris, which is a cylindrate, which is also carrying lamine, expressing lamine. This lamine also carries here this LNDR motif. So this is a very, very old motif, and obviously this is a guess that this might be not really a mutation which will be tolerated by the patient. Well, just to recall that we are the separation of these different the tree of life here shown here. We are talking about a separation of these lines for several hundreds of millions of years. So this might be a very conserved motif which is not allowed to be changed by mutation. Well, what came out? We put in the cDNA into cell cultures, and this is what you can see here. This is the wild type. The other one at the top are controls, and the wild type is forming the filaments. So this is very bright, this GFP tagged desmine. And what you see here is the mutation N116S. So you see these dots here indicating that there is not a filament, but they are protein aggregates. So we purified the protein, and what you can see here is the mutation, the protein, the isolated protein also forms aggregates, not filaments as the wild type. And these are other mutations we tested in addition and published some years before. So the isolated protein and in the surrounding of a cell, the protein doesn't form really filaments. And this is also proven by filament formation. You see here the wild type in blue, in green. This is specific viscosity over time, and you see the mutation here in red. So this shows you clearly that the filament formation is severely affected by that mutation. These are mixtures of wild type and mutant. So we had all these indications that there might be some pathogenic effect by that mutation, but we couldn't do any co-segregation because the family had no other member with that phenotype. But when we had the explanted heart from that patient, we found the aggregates were also occurring here in the patient's heart. This is an anti-desmin staining, and you can see these protein aggregates can be found here. And unfortunately, what was not known before, this was also found in the skeletal muscle. So this patient was confronted. We were confronted, to be honest, with a severe clinical case of ARVC. But during genetic counseling, we had to open up another field because this patient might also be confronted with a skeletal muscle disease in future, and this was part of the genetic counseling. Coming back to that pedigree, why did we find this? Again, this is a de novo mutation. The parents were not carrying that mutation, and this was really a highlight for us. We could exclude that this mutation is also present in the sister of the index patient, and she's doing fine, and she just gave birth to a child. Coming to the third example, which is shows you how strong a panel, without looking strongly for candidate genes, how strong a panel sequencing approach might be to find out what is really going on. Look at this pedigree. You see that this pedigree obviously indicates there must be something in the families. There must be some genetic case here. We were confronted with this patient here. She is a young girl, and she is evaluated for high urgency heart transplantation in our center, and she carries an RBM20 mutation, which is in our hands a pathogenic mutation. The mother is also carrier of that mutation, but also carries a lamin mutation, and this part of the family was diagnosed by a different laboratory, whereas the very left part of that family was diagnosed by another laboratory, and they were looking for arrhythmias because this patient here survived a sudden cardiac death, and she lost her first child, an adult daughter, by sudden cardiac death. And they diagnosed a long QT syndrome, and they found a potassium channel mutation, which is known and is a disease-related mutation. Well, she survived, but she was reanimated for a long time, and she's now sitting in a wheelchair, and she was blinded because she received an ischemic brain damage, which caused these neurological problems now. Well, the family was completely, when she came to our hospital, they were really desperate because they didn't know what is now true, what is valid, and what is relevant, what is not relevant, because this person here was pregnant again, and she was expecting that the genetic testing, and she's also a carrier of the mutation, and carries also an ICD for defibrillation. And what you can see here, that in the meantime we have done panel sequencing and we found out what is really going on in the family. So they are affected by two different genes, the potassium channel, the RBM20, and we know from the lamin mutation here that this is probably not a disease-related mutation, because in that pedigree, in this part of the pedigree, there is no cardiomyopathy present and detectable. So we are quite sure that this can be ruled out as a dominant pathogenic effect. Well, the RBM20 mutation, why are we sure that this is really a disease-related mutation? Well, we found in the RS domain, which is also highly conservative among different species, we found that there is a specific residue which is present, which cannot be exchanged. We had before a serine residue exchanged by a cysteine residue. This was affecting another family. And in the meantime, we have some evidence that this is indeed a phosphorylation site, and the serine is essential. No other amino acid is tolerated at that position. Well, the advantage if you have the explanted hearts is you can look for splice defects, and RBM20 is a splice factor causing around about 30 different known missplicing products in the myocardium, and one of them is the ryanodine receptor 2, which is also a mutation-carrying gene for CPVT, catecholaminergic polymorph-ventricular tachycardia, which is not a cardiomyopathy but a rhythm disease. And remarkably, there's a splice defect which is caused by the RBM20 gene, and the mutation causes an integration of a novel exon. And this can be shown here. When you have the mRNA and you have the myocardium, you can easily see that this is highly significantly different, the amount of messenger RNA carrying this exon in comparison to controls. So there is evidence that there is a macular phenotype in this patient, and the mutation with the exchange of the protein, the serine against protein, will cause also the same problem. And what you see here and on this slide again are the cysteine variants, and the cysteine variants cause a distribution defect, so the protein is not transported in the nucleus, and therefore this protein cannot, of course, be part of the splicing process. So the mutant protein is not anymore able to work there. So with other words, having done all these different experiments in addition and looking for panel sequencing, we came to the conclusion that indeed this mutation has to be classified as a class 5, a pathogenic mutation, which is now part of the genetic counseling in that family. So coming to the fourth example, this is a phenotype-genotype relationship which is remarkable because this is affecting a family by a phospholamban mutation, which is a well-known mutation in the Netherlands, but also present in Germany. And what is remarkable with this mutation is it's a class 5 mutation, so it's a pathogenic mutation, there's no doubt, with an incomplete penetrance, by the way. But what you can see here from the Netherlands, the data from the Netherlands, because they have a huge amount of these carriers, 60% of them develop a dilated cardiomyopathy phenotype and 30% develop a ARVC phenotype. So two different types of cardiomyopathies caused or associated with the same mutation. So the reasons why this or the other phenotype is expressed is not precisely known. So this is also important to know when you have some risk factors. This is excellent work from the colleagues from Groningen in the Netherlands. You see that if you have some risk factors and some additional risk factors, you might have the cumulative probability of event-free survival will drop. So what was the meaning for that, for this mutation in that family? How important was that for the family? Well, what you see here on the left side, the total artificial heart was put in. She was a very slim person. A classical total artificial heart didn't fit in. and she was implanted due to a right ventricular cardiomyopathy. But what about the sister? Well, she was a kind of borderline, and our cardiologists were believing that she is not having that phenotype, and she allowed testing, predictive testing after genetic counseling, and unfortunately she is a carrier of that mutation. And the idea behind that was basically, if she is not a carrier and she has some evidence in the late 50s that she is indeed not a carrier of that mutation, well, the daughters of her will not be confronted with that situation and should not be worried about that familial disease. And this is the last example, which is also a remarkable example which we are confronted with when the index patient came to us asking for a genetic diagnosis concerning ARVC. And what was the family history behind that? Well, the mother lost within 4 months their adult sons by sudden cardiac death. They had some slight symptoms of a cardiac disease, some arrhythmia, something which should be checked by a cardiologist and was checked by a cardiologist. But they had almost no chance, as we know today, because this is the background for this phenotype here is a very aggressive mutation, which is known from Newfoundland, from Canada. Well, within the same year, the cousin of the two died also by sudden cardiac death. A classical case while playing tennis. He died during sports, and he could not be reanimated. And this case here was transferred for autopsy in the forensics department in a certain country in Germany, and they opened the chest and looked for the phenotype, and they couldn't find any kind of morphological changes in the heart. So in other words, an ARVC, and you have seen an ARVC heart before, was not found in this case. Well, when he died, he was transferred to the same department, and they identified an ARVC phenotype. But later on, we found that there were some mistakes within the autopsy, and he was, without doubt, he was a carrier. He was also had a phenotype of an ARVC. Well, the mutation we identified were almost at the same time published when we received the contact to the family. And you see here in the headline, it's a fully penetrant mutation, and there's a huge experimental evidence behind that this variant is indeed the mutation which is causing all these phenotypes. And you can see here, this is the predicted structure of that protein which might be affected. And we were believing, due to the text, to the introduction of that paper, that this is an endemic effect in the peninsula of Labrador, which it is not. So this is the genotype of the index patient. We found, we checked different genes. In addition, we found some additional variants, but the leading example, the leading mutation is this one here, the S358L in TMEM43. Well, the Canadians have huge experience with them, so this is taken from an older paper, but Dr. Hodgkinson from Newfoundland has recently published novel data, but this shows you directly what happens here. So you're losing more than 50% of the patients, of the male patients by sudden cardiac death, aged roundabout in the early 40s. This is a remarkable difference between the genders, as you can see on the right side, because the Kaplan-Meier curve shows you that the female carriers have a realistic chance to reach normal life expectancy. Well, coming back to that story. To our pedigree, you see the computer algorithms, and you cannot rely on that. As you can see here, predicted tolerated in that algorithm is a disaster for a fully penetrant mutation. And what you see here is the genotype we identified in the parents, and where was it possible to do that? Well, because we received some tissue samples which were kind of leftover samples from the autopsy, and the mother managed to rescue them from destruction. And what you see here is in the middle, she was also able to rescue a sample from the autopsy from this forensics department that when we managed to establish a Sanger sequencing of that, we found that he was not obviously appeared not to be a carrier of that mutation. But when we received the report of the forensics department, we realized that obviously the name was wrong and the date of death was wrong, and then we did we looked for some genetic markers between mother and this sample here, and we realized this is obviously not the correct sample which belongs to that family. Well, this is a problem with forensics because the colleagues from forensics are focusing normally on criminal activities, and they are not in charge of looking for the pathology, and this is of course a problem in several countries, especially also in Germany. Coming back to that story, at the same time, colleagues from us in Copenhagen also identified mutation carriers, and the question was, well, what has this to do with an endemic mutation on Newfoundland? Well, the answer came when we made a haplotype analysis, and this is shown here. As you can see here, the TMM43 and the chromosomal markers here, upstream and downstream, and you can see in gray the size of the PCR products which can be received from the fragments we can receive from that. And obviously this shows you clearly that the mutation carriers are all belonging together. They are carriers of the same haplotype. So in other words, the German family, the Danish families, the Canadian families, they all share the same haplotype. And one question was, how old is this founder? How old is this mutation? And the complete surprise for us was the age of that mutation is 1,300 to 1,500 years old. Well, just to recall that, this is the Western Europe map from the Middle Age. So Charles the Great here shown on the left side, who was buried in Aachen. Well, Charles was not living at that time when the founder of that mutation was obviously present in Europe. So in other words, it's a mutation which was exported from Europe to the new countries, and this has a remarkable clinical meaning for the carriers. As you can see here, this is also excellent data from the Canadian colleagues because they have all these patients, and what you can see here is what happens if you put in ICD as a prophylaxis, and what happens if you wait until you have a ventricular tachycardia. You can easily see that this has a remarkable effect on the survival of the patients. In other words, the knowledge of this mutation saves lives. And even this is an old mutation and a rare mutation. It should be considered in every ARVC case you are confronted with. Well, in the meantime, we know that unfortunately 3 of the 4 kids in that family are also carriers, so both of these male carriers, or all 3 carriers are under control for regular checkups in our hospital. So they were checked when they were around about 10 to 12 years old. So basically, what is the practical message behind that? Well, you see very small here at the very left top of this slide, you see the pedigree again, and we were really believing that this mutation was a novel mutation, was a spontaneous de novo mutation here in Germany, and has nothing to do with the Canadians. But from the haplotype, we know they are all related. So in other words, the first one in that family pedigree, which was identified, identified by the index patient, by a woman in her 70s now, could not be the first one. In other words, there must be others out there in Europe who are carriers of that mutation. And what is really amazing for us, was really amazing for us. You see here the map where I'm just currently sitting here in Bad Oeynhausen in Germany. This is shown here in the middle. And the family was by chance coming up from all these villages surrounding our heart center. So we are probably surrounded by other family members who might be carriers of that mutation, and obviously this should be checked in every ARVC case, and because you cannot know, you have no chance to know before what you are confronted with, it's highly recommended to use a broad spectrum of genes for NGS sequencing. And this is where I end up. I would like to sum up the webinar. So I hope you have seen that molecular genetics is of impact for patients with severe forms of cardiomyopathies, and it's highly recommended to do that. However, its impact cannot be predicted at the beginning of the molecular analysis, and we found some discreping data between pathology and for myocarditis and genetics. Without NGS technology, the genetic analysis of cardiomyopathies does not make sense. So it does definitely make sense to sequence a broad spectrum of genes just to find out what is really going on in that family. There's no clear relationship between genotype and cardiological phenotype. Be aware of that. You should be skeptical just for the phenotyping. Sudden cardiac death should be thoroughly investigated by an interdisciplinary team. Unfortunately, this is not a routine procedure in most countries, I guess, for the forensics. And finally, cardiogenetics using NGS panel sequencing is a powerful tool, as you hopefully have seen. However, some pathogenetic relevant conditions might not be detectable, as, for instance, these deletions, as I mentioned before. So this is where I end up with my presentation. I hope you had some impression of the relevance of this genotyping, and thank you for listening.